Hi, cool. So thanks for that introduction, and also thanks to the organizers for this opportunity to speak to you all today. And thanks to you watching for attending. So the somewhat shorter title for my talk today is Feeding Babies Biome. And more specifically, what I'm going to talk about, I'm going to start off by showing you some of our data on the highly varied composition of the early infant gut microbiome and the profound effect that breastfeeding appears to have on the microbiota composition at this age. And then I'm going to zoom in to a question about a key component of breast milk, the human milk oligosaccharides. Specifically asking the question of whether the natural variation in breast milk HMO content that we see across different mothers might explain some of the variation that we see in gut microbiomes across different breastfed infants. And the data I'll show are from the KOALA Birth Cohort study. This is a big birth cohort in the Netherlands with a lot of questionnaire data and also a funny looking koala for a logo. And as well as that, though, in the KOALA study, we have fecal samples from over 1,000 infants taken at 1 month of age, from which we profiled the microbiota composition. And then from the 220 of the mothers of those infants, we also have breast milk samples from the same time point. And we use liquid chromatography-mass spectrometry to quantify 15 of some of the most abundant HMOs. But before zooming into that HMO data, first we'll have a look at all of the microbiota data and its associations with breastfeeding and a couple of other important determinants. So here what you can see, this is a circular bar chart showing all 1,000 of the infant fecal microbiota samples sorted by similarity. And you can see clearly that there are quite some different compositions present, some dominated by Bifidobacterium in light blue, some by Bacteroides in light green, some even by Enterobacteriaceae in this darker blue, and many then with a mixture of these 3 taxa and several other taxa. And from our statistical analyses, we found in particular that 3 exposures were each having big independent associations with the microbiota at this age. Firstly, C-section. We heard a little bit about C-section already today, but the C-section born infants in our data set, they're highlighted in purple on the PCA plot of the microbiota data. And you see these infants clustering mainly on the left side. And in fact, the position of the samples on the PCA is also what orders the samples on the bar chart. So you see the C-section born infants on the top left of the bar chart as well. And then you can spot that characteristic lack of the light green Bacteroides that a lot of these C-section born infants characteristically have. So yeah, birth mode shows profound associations with microbiota composition in our data. And if we now color the infants on the same PCA by the amount of breastfeeding that the infant received, we can see this time a clear gradient going from top to bottom with the more blue points of the more formula-fed infants at the top of both plots, separated from the whiter colored, exclusively breastfed points infants at the bottom of the plots. But I'll tell you a bit more about the breastfeeding associations on the next slide, because first I want to take a little brief sidetrack to mention a couple of our results on antimicrobial exposures. So a few infants were directly prescribed antibiotics during the first month of life, and they're highlighted in orange on the PCA plot here. And you might expect this early direct antibiotic exposure was associated with quite large differences in microbiota composition. But also, there's been some speculation in prior literature that 2 other antimicrobial exposures might also influence the infant bacterial microbiota, namely antibiotics given to the mother during pregnancy and antifungals given directly to infants. And both of these exposures, they were quite common in our data set. However, we didn't see any obvious effects of these on the infant bacterial microbiota at this time point. And then as a last note for this slide, we also found other interesting associations with a variety of other exposures, such as home birth, as was mentioned also a bit earlier, and the presence of pets and siblings as well. But I don't have time, sadly, to talk about all of those things today, because we're going to zoom in and have a little bit of a closer look at what breastfeeding was associated with in the 1-month microbiome. And the plot you see here, this can be read a little bit like a heat map. with each node on the tree structure showing the strength and direction of each taxon's statistical association with the amount of breastfeeding that the infant received. And the concentric rings of nodes represent aggregation of the amplicon sequences at different ranks of taxonomic classification, from phyla around the middle ring all the way out to individual ASVs around the outer ring. And breastfeeding was associated with a lot of these taxa. Some key examples include, of course, a higher relative abundance of Bifidobacterium, a lower relative abundance of Bacteroides, and a lower relative abundance of many Clostridia, amongst many other associations with the amount of breastfeeding. But don't get the wrong idea, as despite all of these associations, the microbiomes of different breastfed infants are not all the same. And this brings us to the question of HMOs. Might variation in breast milk HMO content help explain some of the still unexplained variation in the microbiomes of different breastfed infants? Firstly, though, why do we suspect that these HMO things are so important for the infant microbiome anyway? What are they? So firstly, they're a major component of breast milk. They're the third largest solid part, but they're not food directly for the infant. Humans don't make enzymes that will metabolize these HMOs, but some members of the infant gut microbiome do, particularly some bifidobacteria, for example. And these HMOs, they're complicated things. They all share the same lactose core, but this core can be extended and decorated in various different ways and combinations to make dozens of different structures. And it appears that these different structures have different functions in the infant gut, not simply acting as just prebiotics, but also doing other things like potentially preventing pathogen adhesion or interacting directly with the innate immune system as it develops. Interesting stuff. Okay, so we measured 15 different HMO structures, some of the most common ones, in 220 mother's milk samples. And you can see that the breast milk HMO compositions are shown here on stacked compositional bar charts. And you can see that the mothers are split into these 4 different groups, with each group showing a very different HMO composition to the other groups. And these groups are corresponding to combinations of the genetically determined Secretor and Lewis groups, which relate to the mother's ability or inability to synthesize particular fucosylated HMOs. And so we suspected that these large differences in HMO production might correspond to substantial differences in breastfed infant microbiota composition. But that's not quite what we saw. In fact, many different infant gut microbiome compositions were observed within each of these HMO genotype groupings. As you can kind of see from the variation on this heat map of microbiota composition that I've added to the slide now. So we saw no statistical associations between microbiome composition and these Secretor-Lewis groupings. And indeed, the prior smaller studies, so looking at the same kind of question, have shown mixed results, with some showing associations with Secretor or Lewis status and some not, like ours. But nevertheless, our story doesn't quite end there, as there is still further HMO variation that isn't driven by these genotype groups, particularly in neutral and sialylated HMOs. And indeed, when we looked in detail for possible associations between individual HMOs and microbiota composition, 2 HMOs stood out as perhaps being interesting. One was LNH, an undecorated neutral HMO, and the other was 6SL, an acidic sialylated HMO. Both were associated with overall microbiome composition, and indeed both were actually associated with Bifidobacterium relative abundance, but in opposite directions. So when a mother had somewhat higher LNH in her breast milk, this was associated with a relatively higher relative abundance of Bifidobacterium in the infant gut, and the higher abundances of 6SL relatively were conversely associated with relatively lower relative abundances of Bifidobacterium. I do want to emphasize at this point that these associations we're spotting here, they're not nearly as like profound or compelling as the effects of breastfeeding versus no breastfeeding. But nonetheless, it's possible that these two may be interesting molecules to prioritize in future HMO research, particularly as relatively little is known so far about their roles in the infant gut. So that already brings me to my final slide, where firstly, I'd like to thank all of the organizations and the people who made this project possible, especially my colleague Martha Endicke, who did a lot of lab work for this big project, and she shares joint first authorship on the paper as well. Before I completely finish though, I'd like to throw in a cheeky advertisement of my R package for microbiome data visualization, that's MicroVis. And I say this because I've tried to make it a bit beginner friendly and partly interactive. And so perhaps it might be helpful to anyone listening who is maybe just starting out on their own microbiome data analysis or something like that. So if that perhaps sounds interesting to you listening, you can find the tutorial website at this link, or just Google it. And with that said, thanks again for listening to my talk, and I look forward to your questions later today. So thank you.